Okay.
Go ahead.
So I'll begin by thanking Vijay and Anjana for putting together a really nice meeting. I think I've heard talks that I would probably not have heard at other meetings I normally go to. So it's been really fun. I've learned a lot from it. And I'd like to also thank them for giving me this opportunity to speak here. So what I'm going to talk about is how we move and shape groups of cells during morphogenesis. And I hope I'll illustrate to you the importance of being morphodynamically and mechanically heterogeneous. We're broadly interested in the lab in looking at what we call morphogenesis, which is just it's a term that refers to processes by which cells, tissues, organs, and organisms acquire the shape or form that's critical to their function. And we particularly focus on the morphogenesis of epithelia, which are essentially layers of cells that are closely bound to one another to form sheets that either cover surfaces or line the insides of organs. And some general rules that govern morphogenesis is that at the cellular scale, it's governed by changes in cell number, cell type, cell shape, cell size, cell position. And these cellular processes are in turn regulated through the interplay between molecules, through gene regulation, gradients of the kind you just heard, signaling protein interactions, but also through mechanics, which includes force generation in living systems through molecules that include adhesion in cytoskeletal networks. And the other thing I think is well appreciated is that to shape tissues, you need integration of information at multiple length scales, starting from molecular to tissue length scales. And so we've basically been using the fruit fly embryo that you already saw in Tim's movies, except that I'm showing you a view that looks at the embryo from its back and from its belly side, rather than a side view that Tim showed. And in these movies, every single nucleus of the embryo is labeled. So you'll see that in the process that makes an embryo become a maggot, there's a lot of juggling around of cells, a lot of deformations of cells, lots of movements of groups of cells. And you'll see some cells are large, like these ones here. Some cells are small. Some cells move collectively. Some cells behave singly. Till at the end of all of these multiple events of deformation, the embryo eventually becomes a maggot and hatches out of its shell. And so we've been interested in looking at how, in this seemingly fluid morphogenesis of the fruit fly embryo, does the embryo maintain a balance between being deformable and maintaining shape? What is the precise nature of the interplay between molecules and forces, and how is information integrated across multiple length scales? And so we really use the embryo as our test bed and focus on looking at different types of morphogenetic movements, just because they give us a handle on the diversity in the ways in which you can generate what I'd call dynamic form, because these forms are, or to build these structures, you need cells to be dynamic. So with three processes that we work on in the lab, and from all of today's talk, I'm just going to focus on one, which we call germ band retraction, during which, as I'll show you in a movie in a minute, you'll see that the germ band, which is this U-shaped structure here, moves from the head end of the fly embryo is here, the tail end of the fly embryo is here, moves from the head end to the tail end, so that the tail, which forms here, is positioned at the end opposite to the head rather than right next to it. You can imagine what happens if this process fails. So I'll just show this to you quickly in a movie. So this is the head end. This is the future tail of the fly embryo, positioned initially at the head end, but moves in that direction to become finally positioned at the tail end of the embryo. And so the work I'm going to actually talk to you about today was done by two talented graduate students, Shuripa Nandi here and Charvi here, and it was actually initiated quite a long time ago, and I'm just putting this picture here because this was, I think, Mike Sheets' last visit to India, and this is Shuripa actually describing the beginnings of this work to him many years ago. Okay, so go back to germ band retraction. So this is what happens in germ band retraction. You've the germ band moving, retracting back to its final position so that the tail end of the germ band is positioned at the tail end. And in this process, you have several other things that happen. This orange tissue here, which is stuck inside the fold of this U-shaped embryo, actually flattens out and becomes exposed on what we call the dorsal side. The anal pads, which are the hind end of the gastrointestinal system in the fly, actually become positioned at the hind end. The hind gut, which is part of the alimentary tract, also becomes positioned at the hind end. And the segments, which you saw beautifully in the last talk, actually become arranged in a linear order along the head-tail axis of the embryo. And many years ago, in the beautiful screens of Yanni Nusslein, Bolhard, and Eric Wieshaus, mutants were identified in which this process fails. And you can see what the consequences of it are. These embryos don't have a straight axis. The head end and the tail end are close to each other. You can imagine the hind gut is also close to where the mouth is. So failure in this process leads to body axis abnormalities and to defective organ positioning. And so it's important for us to try and get a handle on how the axis straightens and whether the straightening of the axis might happen the same way across organisms or not. And so what we decided to do is to just look at what happens during germ band retraction. And what I'm showing you here is a view of the embryo looking at the dorsal side. And I'm going to focus on two tissues here. This entire tissue here with small cells is called the germ band. This tissue here with the large cells is called the amnioserosa. I'm going to focus on this red cluster of cells here just because it's a group of cells that moves the farthest. Or that sort of, it's like a marker for us, so we know how far it's gone. And what you'll see when I play this movie is that the tissue with the large cells, which sort of covers the red heart-shaped structure as if it were a wheel, gradually unfolds and spreads out onto the dorsal surface. The red heart-shaped structure emerges from underneath it, but at the same time, it sort of reaches the right-hand side of the screen, which is the tail end. And if you look at this, look at the configuration of the tissue in what we call a sagittal view, which is the kind of view that Tim was showing us, you'll see that the amnioserosa, these large cells, form a fold that lies over this red-shaped structure, which here is shown in green. And with time, those folds sort of flatten out till at the end you have both of these tissues arranged as if they were one sheet. That's just the first part of retraction. In the second part of retraction, and I'm going to play this from the beginning because it's playing too slowly. So this is the end of the first movie here. Now, if you watch what happens here, the cells that were in red in the previous movie that are now here in magenta, you'll see that the magenta region actually moves away from this tissue that has large cells, and a non-magenta field of cells comes and occupies the space in between. So what happens in the second part of this retraction phase is that the caudal end of the germ band moves away from the amnioserosa, and other cell fields take its place. And so we've been trying to ask some very simple questions. How does an epithelial sheet unfold? How do cell fields within that epithelial sheet become positioned at distinct locations? What governs the kinetics of retraction? So these are some of the questions we set out to ask, and used a combination of methodologies to try and address that. But I should remember to mention that a lot of this work was sort of facilitated by really productive collaboration with Vijay, who's right here, and Mandar Inamdar from IIT Bombay. And so the first thing we set out to ask was, well, basically, it looks very complex. Can we parse this process into spaces and phases? And I already showed you two parts to retraction. So in the first part of retraction, the thing that hits you in the eye is that the shapes of the cells in this part, the germ band, are not all identical. And so you can begin to see with time that you build up heterogeneities in cell shapes with some cells close to the midline. So here at the bottom, we're looking at just half of the structure here. The cells close to the midline are large, cells a little bit away from the midline appear to be stretched and narrow. If you look at the second part of retraction, when I said that the magenta colored structure actually moves away from the amnioserosa, you will see that the original heart-shaped structure becomes rectangular and then actually inverts itself so that the pointed end of the heart is at this end rather than at this end. And then it's almost as if this heart-shaped structure pinches itself away and moves backward. And as a consequence, what you'll also see is that cell fields that were oriented along this axis, right, actually eventually become oriented along this axis, meaning horizontal sort of becomes vertical. So these are two gross features that we noticed. The other feature that we noticed, and this is again helped by Mandar and Vijay, is that the speed of this retraction process, figured out using particle imaging velocimetry, is temporally regulated. So the germ band retracts. So there's a phase when the germ band is just stalled, which is followed by a phase when the velocity of retraction gradually increases to reach a peak velocity before it actually exhibits a fall. So the speed is regulated. So there are phases, there are spaces. And so given that the whole germ band is retracting towards the posterior end, we also wanted to get a feel for how, what the local morphodynamics is. And again, with Mandar's help, by making what we call moving windows, also attempted to look at what the local morphodynamic features of the germ band are. And so what I'd show you now is a high resolution movie of what the outlines of the cells look like as the germ band retracts. And what you will have seen in that circle as it came up is a radially organized structure that looks a little bit like an onion, as as it comes out, and you'll see that that anisotropy that I spoke to you about. And if you actually quantify this, quantify cell shapes, what you'll see as a function of space and time is that as you go from the midline towards the periphery, cells look more stretched. This is evident in their shape anisotropy, shown here in blue. You're going from the center towards the periphery. You can also see it in the aspect ratios of these cells that rises as you go from the center towards the periphery. The other thing you notice is that the cells at the periphery, this yellow zone here, also appear to be more aligned. So there's more coherence in the cells at the periphery compared to the cells in the center. So what we're seeing in this part of the germ band is that there's a radially organized morphodynamic gradient that's characterized by shape anisotropy, nematic order. And what I've not told you is also a gradient of intercalation that we've sort of used cell-sidedness as a proxy for. And if you look at the shape index here, you'll see that the cells at the center have shape indices close to 3.8, but as you go away from the center to the periphery, those shape indices are much larger. And so this also led us to suggest that there's likely a gradient of fluidity with cells at the center being relatively more solid compared to cells at the periphery. And again, with Mandar's help, we also measured the shear strain rate along this axis. And you can see a small but significant increase in shear strain rate to go from center to periphery. If you look at what happens in the second part of germ band retraction, you both convergence and extension. If you just follow these two dark black dots, you see that they're coming close to each other. But if you look at the sort of magenta color, you'll see that the magenta is actually moving away. So there's extension along this axis, convergence along this axis, except that this convergence and extension is happening between tissues rather than between individual cells. So you can see that separates the amnioserosa away from the caudal end of the germ band and allows other cell fields to occupy the space in between. And then if you look at, so there's all this is also accompanied by a sort of buildup in the diagonal shear as this as the process goes on. So we've two sets of events happening. One that's associated, the early part that's associated with a radial morphodynamic and mechanical gradient, and the second part that's associated with what we call a multi-tissue T1 transition. And so next question we wanted to ask is how are these gradients and transitions achieved? And we just focused on the usual suspects, the actin cytoskeleton. And here you can see what happens to myosin. This frame keeps shifting back to here so that you can see it. And you can see this onion-shaped structure is actually fenced by supracellular actin cables, whereas the final detachment phase where this red bit moves away is mediated by that T1's transition that results from the constriction of the cable towards the end. So just pointing it out to you again. So you can see the emergence of cables, one at the periphery first, and then concentric cables that surround this onion. Then a cable that forms here that constricts till at some point you'll see that this bit actually pinches off and moves backwards. And so we have two types of cables. The cables that form around this radially organized structure actually do not constrict, and their position distance from the midline, for example, is conserved. These cables here that mediate the T1 tissue scale T1 transition actually constricts. Nonetheless, both of these cables are tensile, and if you use lasers to ablate them, you'll see that both of them recoil. But if you look at the features of that recoil, you'll see that there are differences, maybe not so much in the recoil velocities as much as their subsequent behavior that likely also tells us that these cables might be different in other interesting ways. And then we were also able to see that this cytoskeletal organization actually nicely recapitulates the cellular organization we see, both with respect to order, nematic order, but also with respect to the anisotropy in both phases. And that sort of led us to ask whether and how the organization of the actin cytoskeleton maintains or contributes to the structure and dynamics during germ band retraction. And the first thing we saw was that if you perturb actomyosin contractility in this part of the germ band, then you don't see such a nice radially organized structure. But what was surprising to us was that the duration of the fast phase was actually reduced and the average speed in the fast phase was increased. This suggested to us that reducing actomyosin contractility affects the early part of retraction by hastening it, but also flattens the gradients in this radially organized structure and promotes cell intercalation across all populations in this structure. And if you think of cell-sidedness as a proxy for the ability of cells to rearrange, then basically what it tells us is that perturbing contractility sort of fluidizes this structure in the early part of retraction. But if you perturb contractility by nicking the cable here, and I won't show you the movie in the interest of time, we see a loss of coherence in that structure. The radial organization, cellular organization is affected. We also increase the shear compared to the unperturbed cable embryos. And we also reduce the symmetry in flow between the right and left sides. I forgot to mention that we nick the cable only on one side. So basically, if we perturb the supracellularity of the cable, we don't have coherence in this sort of radially organized structure. We lose nematic order. We reduce bilateral flow symmetry. But what we do is that we enhance retraction velocity and shear propensity. And so I think what that told us is that actomyosin cables act at this early phase as brakes or fences to maintain coherence, brakes to regulate speed, and disrupting them causes a loss of both coherence and then sort of runaway dynamics. Perturbing actomyosin contractility in the second phase also disrupts and delays detachment. So rather than hastening detachment, it slows down detachment and it alters the organization and curvatures of the germ band. And so eventually, I'd like to just point out that if you perturb actomyosin contractility, you don't position the organs the right place. So these are unperturbed embryos. This is the end of the gastrointestinal tract, which you can see is fairly nicely confined to a sort of smallish region here. You perturb contractility, you have an expansive hind end. And it's also misshapen. And we don't yet know how this affects the function of the gastrointestinal tract, and we're sort of trying to work that out. But here are some 3D renderings of the hind end of the embryo that allow you to see that in a normal scenario, this is what the hind end looks like in multiple views. And if you perturb contractility, misshapen and mispositioned. So I think in the interest of time, I'm going to stop there. But I just want to say that we've begun to look at these structures in 3 dimensions, and we see that it's not just the apical shapes that are patterned, but also the heights of the cells that are patterned, and there's a central to lateral gradient in height as well, as you can see here. And what I won't be able to tell you is that this patterning, 3D patterning of shapes, actually dependent on adhesion anisotropies that are due to differences in the way integrant-substrate interactions are patterned along this axis. And I will just end by saying that that future work focuses on how actomyosin pools and heterogeneities in integrant ECM interaction influence the morphodynamics and kinetics of the germ band. I'll stop and take questions. So what I've basically told you is that germ band retraction is not a single cell behavior process, but it's associated with heterogeneities and morphodynamic and mechanical properties of cell cohorts in time and in space. And we think that these properties influence material properties of cells, and through it influence the kinetics of retraction. Shown that the spatiotemporal regulation of cytoskeletal organization underlies these heterogeneities. And what I didn't have time to show you is that patterned and dynamic cell-substrate interactions govern both organization, morphodynamics, tissue topology, and kinematics. And I'll stop there and take questions.
Thank you very much. Any comment or questions?
Thank you very much. So one thing you didn't discuss is that, of course, underneath is the nerve cord, which is also retracting.
What do you think? What do I think is underneath?
No. So underneath is the nerve cord, which is also retracting at the same time.
Beneath this radially organized.
Yeah. And so does that play any role in, because it sort of has its own independent retraction, but it's also linked. So does the behavior of the nerve cord interact with that, and vice versa?
Part of it, the medial part of it, lies on top of the visceral mesoderm. The lateral part of it just has loose cells, is the substrate whose identity we've still not worked out. But we've reason to believe that the medial substrate is possibly stiffer than the lateral substrate. And there are ways in which we're trying to tackle that.
Actually, that was beautiful. I'm just curious about whether all of this reorganization is remodeling alone or any addition of new mass, either in terms of new cells or cell growth itself. Is there any addition of new mass, as in cell growth or addition of new cells via replication, that also contributes to this remodeling? Or is it just the remodeling of the cells and the cell shape that is triggering all of the movement?
So what was the first?
Mass. Like, yeah, is there growth? Is there cell growth?
All of that.
Huh?
Division numbers?
Yeah.
Okay, so that's a good question. So we have looked at that. So we know that proliferation is sort of very localized to the rim of this structure. There is no proliferation in the sort of labeled cells that I showed you. So there's actually no cell addition there. But what it actually is, so the growth is actually, the growth is actually coming from exposure of tucked in cells. So you can think of this structure as a bent hockey stick, and the cells in the bend are actually coming on top like that. But the structure is also, symmetric across the midline, and there are some cells that are tucked at the midline as well, and these are coming up like that and exposing it. And what we've been able to show is when you interfere with interactions with the substrate, these cells that sort of emerge onto the surface from underneath in the midline actually don't emerge, but instead the cells that are on the surface cave in and go down. So it's really a series of unfolding morphogenetic movements that drive it.
Could you just say how you plan to look at the ECM and the integrants during this process?
Speak a little louder.
How do you plan on doing that? Looking at ECM and integrants in this process? You said that's where you're going, right? How are you planning on looking at integrants and ECM in this process?
I did.
How are you going to do that?
Oh. How am I staining it?
Well, yeah.
Studying it,
essentially.
okay.
Yeah, tell me how you,
Okay,
it's hard to imagine
we're
this whole
basically
complexity.
looking at it genetically using a whole host of perturbations, including function modifying as well as reducing the levels. With respect to looking at material properties of this substrate, because the substrate is sort of basal, we can't easily look at traction forces and that sort of thing. But what we are trying to do is to bleach small regions of extracellular matrix proteins tagged to GFP and look at how those bleached spots actually move. A little bit like putting some beads underneath, but we can't sort of get beads to that place. So I think the way we're thinking about it is to bleach, let's say, laminin tag to GFP, and then analyze the movement of those bleached spots before recovery. Okay.
Okay, thank you very much, Maithreyi.